I'm going to talk about what we have been doing on drug discovery and also how LLM can actually contribute to this drug discovery research as well as healthcare industry overall. My talk will be a little bit different flavor. The rest of the talk today I think is about genomics, but my talk does not have much of genomics. I'm going to talk about drug discovery and also LLM. The first half will be about the drug discovery and the second half will be about LLM. Okay, so some of you might have seen this slide already because I see some of the familiar faces because I use this slide several times. So 이름's law. The irum is not the name of a person. Actual name is Moore. Moore's law. Moore's law is quite well known. So, but this is the reverse of the Moore's law to stress that the decreasing productivity in the drug discovery sector. The y-axis is about the log scale of the number of new drugs approved per $1 billion R&D cost is decreasing. Today it's less than one drug after more than 1 billion R&D. Right? It takes about more than 10 years. People getting worried about the low productivity because it's getting increasingly fast getting a red ocean, the drug development sector overall. So the people turn their attention to AI in the hope that the AI can have increase the productivity. So AI might have us getting out of this, difficulty. So my lab, me and my students in Korea University, Computer Science Department, have been working on this drug discovery by applying AI technology to drug discovery. So we have been participating in, because we are not the drug discovery domain experts, no more biologists. So trying to understand what's the real unmet needs in the domain, we participate in the international challenges. Some of the challenges is about drug combination, synergy prediction, and also cancer proteome prediction given the DNA and RNA information, how you can translate that information to proteomics data. And also discovering drugs targeting more than one pathway, etc. And those three are about the LLMs. It's about given the paper which LLM or AI can answer the question related to the paper more accurately. Those are the challenges. Yeah. We have been competing with some of the well-known in the academia industry here. And also the Janssen, that's one of the big name in the pharmaceutical sector, and also Google in the big name in the LLM. We have been achieving a pretty good standing for the 7 years over the past 7 years. So, sorry about the Korean, I didn't expect I'm going to speak in English today, so as I didn't make this English version, sorry about this. But anyway, we have been pretty good experience in the challenges and applying this AI technology to the biotech. We collaborate in collaboration with the biotech, and then the result was not too bad. So I became serious to found a company called ANNI Sciences about 3 years ago. This is an AI-based drug discovery company. And this, the AI drug discovery platform we are using in the ANNI Sciences is based upon the key technology that we have developed in the international competition. Put them together into two components, Agent Insight and Agent Can-Tell. Agent Insight is the LLM-based hypothesis generation model. So all science starts from hypothesis, right? Typically by doing search on the literature. So LLM can help on this. So we developed an LLM for hypothesis generation. And also Can-Tell is another generative AI that actually designed the drug structure. So we use this too. And briefly introduce the two technology. The first one, Insight, is a biomedical LLM. The foundation technology is basically the experience from BioHealth. The BioHealth is the first language model applied to the first language model applied to biomedical domain. Because it was the first model that has a good citation compared to the other competing language models. So this is the encoder part of the transform. If you're familiar with the transform architecture, the part is the encoder part. Trying to understand the context of the text. The decoder part of the transformer is generative. Is the foundation of the whole generative AI today. This was the encoder part, and then using the decoder part, recently we developed the biomedical domain specific LLM, which we named it as a meerkat. I'm going to explain this in the later half of the talk. More on this. And this LLM can help primarily in pharmaceutical company because it's a biomedical specific LLM, and then also healthcare sector as well. So pharmaceutical sector, we can use this to generate hypotheses. In drug development, the two key hypotheses is mechanism of disease. What's the cause of the disease? That's the one key hypothesis. Second hypothesis, how we can inhibit the disease-causing pathway. That's the target hypothesis. What kind of target protein that we can, choose as a therapeutic target to cure the disease. There's two hypotheses. We can use this LLM to generate those such hypotheses. Patient's EMR record, and then when patients come into the doctor's office, the LLM can also already understand the history of the patients, and then the family history of the patient, and also the what kind of drugs this patient has been treated over the history of the therapy. And the other insight is also not only based on the LLM, also data-driven target discovery as well. So basically using differentials of case and control omics, and use the to predict the perturbations. In this case, the genes. Which genes can induce the differences between case and control omics data using this model. This is the previous version of the omics. At that time we only used the transcriptome data. So using the transcriptome differential, we map this differential induced by the particular drug into the same embedding space, and by inputting this expression of the case and control, we rank the drugs that can induce these changes in the case and control. We apply this to the CADASIL. CADASIL is the rare disease in brain. We apply this technique to identify the FDA-approved drugs that can alleviate the phenotypes, the disease phenotypes. And the second generative AI is a drug designer. And the two key features is using the reaction template, fragment generation. So basically generate this part, this is the protein structure pocket, and then this is drug. Drug that can fit into the pocket, that's a good drug, right? High binding affinity. So that the inactivate this protein's activity. So generating based upon the reaction template to guarantee the synthesizability. And also another one is direct preference optimization. Do the multi-parameter optimization is important process to the lead optimization process, and then multi-parameter at the same time we can optimize through the DPO. So using this generative AI for drug design, it has been quite successful. We are developing 14 different pipelines. It's still only like one and 1 year and 4 months since we synthesized the first molecule. It was quite successful. This is one example, the JAK3 inhibitor. It took only 2 months, less than about, this is less than 20K US dollar. So it's been quite successful. We obtained the lead of 2 nanomolar IC50 activity. So another example. The portfolio, we are focusing on different category of drugs. There's D-ubiquitinase inhibitors, and also KRAS portfolio, and selected targets, and some of the resistant cancer targets. So I'm going to talk briefly about the source one and path target X, and then move on to the LLM side. The KRAS is very well-known oncoprotein. So recently, for over 30 to 40 years, KRAS known to be an undruggable target because the surface does not have the pocket that the small molecule can bind to. But recently, people discover some of the small pockets on the mutation-specific protein conformation. And also, people working on the indirect inhibition of the KRAS by inhibiting the protein called SOS1. SOS1 is a switch that turns KRAS pathway off to on. So by inhibiting SOS1, so indirectly inhibiting the KRAS pathway gets turned on. So that's one pan-KRAS mechanism. Another one is a direct inhibitor of the RAS. And I'm going to talk briefly about the source one, which is the pan-KRAS approach indirectly inhibiting the KRAS pathway. Well, this is the all drug development. You need to prove that your drug is more potent than competitors, right? So you typically do a head-to-head comparison. This one is the most advanced one in the market. It's currently in the phase 1 clinical trial. And this is the lower the better, the number lower the better. And typically about 2 times more potency in the drug that we developed. The number 12 means is a 12th compound that we synthesized. Typical drug development, it takes more than 200 or 300 molecules synthesized before you can actually identify lead. But thanks to the AI, we were lucky to identify the molecule in the 12th synthesis. And another one is that the market leader does not reach to the 100% regression, but our molecule actually reached to close to 100% regression. So it's quite meaningful results. And also combination synergy is proven by combining with the sotorasib. Sotorasib is a KRAS G12C mutation-specific inhibitor, recently approved by FDA. And the synergy has been validated through the in vitro assay. And trimetinib is the MEG inhibitor. The MEG is the downstream of the KRAS. So combining the KRAS, pan-KRAS inhibitor and the downstream of the KRAS also show the synergy. And osimertinib is the upstream of the KRAS, EGFR inhibitor. By combining the source one and EGFR also shows some synergy. So we are working on this to further development. Another one is a PAP inhibitor. So the PAP therapy is a standard therapy for the BRCA deficient cancers. Right? So, but the problem is not all patients with the BRCA mutation respond to the PAP therapy. Even if a patient responds to PAP therapy, it develops some resistant mechanism after several months. So there are big unmet needs. So to overcome this unmet need, we identify new target X that can be synergistic to PAP inhibitor. So by combining these two, make one drug having two warhead, one targeting PAP, another having warhead targeting target X. It shows some synergistic effect. Importantly, those cell lines are PAP-resistant cell lines, and then the first generation, second generation PAP inhibitor does not show some effect, but ours actually reaches to 100% regression. Okay, so moving on to the LLM story. The target discovery is one of the key hypotheses in drug development. The first thing that we need to do, identify target. Once we identify target, then we obtain the structure of the protein, and then do the research on the structure, and identify, predict the pocket, and then design the drug that can fit into the pocket. Okay? So the first step is important. Target discovery is important. If the target is not correctly selected, you spend a lot of money, and you typically more than $1 billion, and typically it takes more than 10 years. And then eventually you will fail. The clinical trial, after clinical trial failure, some of the research shows that more than 50% of the reason of the clinical failure is because of the target selection, incorrect target selection, right? So target selection is very important. Target selection research is starting from the literature search, typically PubMed. PubMed is the Google for the biomedical experts. But more than 3,000 papers published every day. Okay? More than 3,000 papers published in the peer-reviewed journals every day, and then added to the PubMed index. So it's already exceeding the human one single researcher's capability. So you need some help from the systems like LLM. So we use the LLM to identify this target X, the novel target that is synergistic to the PAP that can help overcome the PAP resistance using LLM. And not only, if you have a capable LLM, it can help not only design the hypothesis, but also various tasks. Right? This is the hypothesis generation scenario, a potential alternative target PAP in treating provocative gene cancer. You know, the LLM can search the recent literatures and knowledge bases, and then, you know, based upon this context, generate some reasoning, and then finally give you some plausible hypothesis. Now, another one is drug discovery. We can use this LLM to analyze the pattern landscape, right? If there is any pattern filed by the competitors, you need to understand in the first place before you start designing drugs, right? And also, even the drug design. So LLM can work as a research assistant, or co-pilot, or agent, whatever the name you call it. It can work as a human assistant. Typically, well-trained research assistant, you can give it high-level tasks, right? The assistant do the strategic planning, how to execute this task, and research on the available tools, you know, which he or she can access to, right? So LLM can work like that. So eventual goal is to make the co-pilot for the drug discovery research, the LLM as a co-pilot for drug discovery research. And in the drug discovery, a lot of tools are being used by the experts. One of them is a simulation, it's a molecular modeling. The LLM can actually understand what kind of tool available and the inputs and the outputs of the tools, and what the results the tool can generate. And by connecting this intermediate together, finally get to the final answer. So in order to develop such domain-specific LLM, at least we need to go through these steps. You can start from some foundation model, right? Like a Llama or this one, that's open source foundation model. They are trained on the general domain text, typically called by the web. It understands the general language, human language, and has some good coverage of the general knowledge. Get this foundation model, and you do another training on the domain-specific corpus, typically the literatures, papers, and then what others? Clinical report, and clinician sky, such like that. And then, to perform the complex the tasks in the medical domain, the LLM has to have some multi-reasoning ability. So, specifically train this ability, we need to have another data set to teach the LLM to do the multi-reasoning. And then, going through this process, we obtain the MURCAT. This is our first LLM, first version of the LLM for domain-specific tasks. And this is our the model, for language model. So this is generated by generative AI, DALL-E 2. It's pretty nice, right? Isn't it? But there's one problem here: hallucination. Only the 3 claw on the left foot and the 4 claws. Normally it has to have 4, but we tried to modify the prompt, but it didn't actually get to there. So we just use this picture. Anyway, pretty nice. So this one, only the 70 billion parameter. No. 7 billion parameter, which is very small. You can run this LLM. If you are doing only the inference, you can run on a high-end PC, gaming PC with the GPU about 3090 RTX GPU. It's pretty accessible. You can run this LLM. What does this mean? You can deploy this LLM in-house, on-premise, in-house. Hospital, the very sensitive data, EMR records, you cannot upload the EMR record to OpenAI to train this. That's impossible, right? So you have to have some on-premise AI. Meaning, the small size LLM can achieve a high score. This is a USMLE score. The passing mark is about, on average, 60 point. But the 7 billion model is 74. The best performing model is so far, the GPT-4 is 82 points, pretty close to here. We are working on a 10 times larger model, 70 billion model now, and we are training on this model, and the very initial result is quite promising. They're already passing the 80 point mark already. So the thing is that how the small model, the low capacity model, can achieve high score. The key observation is that it's more important. The model size is important, obviously, but more important is that what kind of data and how you train the model. So in order to achieve a high score in the USMLE, the medical school students in the US, to pass this exam, they have to study, right? Using one material, perhaps the medical textbooks, and also the exam preparation books available, right? So we collect those, the textbooks and exam preparation manuals, and 18 different textbooks. And from this textbook, not just let the LLM read the word by word, but we generate the USMLE style questions from this textbook, and then generate the chain of thought reasoning, step-by-step reasoning, how this problem has to be addressed. And we train this chain of thought reasoning data to the LLM, and that was quite effective. So those are the textbooks. And this is the one section of textbook, and then this generated question from this textbook. So we use the state-of-the-art LLM, which is GPT-4, obviously. So we use GPT-4, give this section to the GPT-4, and then show 3 examples, USMLE example, and then GPT-4. Let GPT-4 generate the questions. And this generates the question. And also, we use GPT-4 to generate the reasoning. how to solve this question step by step. And using this reasoning data, we trained LLM, and it was quite effective strategy. So here's the result. The same class models, the most advanced one before Meerkat was Meditron. It's by ETH Swiss. It was a 52 score, not passing the passing score 60 point, but the model achieved a pretty good score here. This score is even better than the 10 times larger model. Okay, the state-of-the-art model, the 70 billion model is about 70, but the 7 billion model 70 is quite promising. Not only the USMLE, but all 13 biomedical benchmark, the score was about 13% better than the GPT 3.5. So here's one example, sorry about this Korean, the USMLE problem. Right? I will test this. So not only just solving the problem, we primarily train using the USMLE style question answering, but we also instruct and tune this LLM on the diverse tasks. One of the tasks is patient consultation. So let the Meerkat work as a doctor, and then let GPT-4 work as a patient. So, and we gave one USMLE question to GPT-4 for reference, and then based upon this question context, GPT-4 role play as a patient, and then ask question to the Meerkat. And Meerkat did not have access to the question context, but based upon its knowledge, respond to the patient question. In this scenario, is a mother called on doctor. She concerned about 10-year-old son. The pain is a mess in his left testicle. And Meerkat kindly asking for another symptoms. And then do the conversation. Finally, you schedule an ultrasound exam at the end. The same case, but in this time, the mother has the lab test results. More detailed results about his son. Her son, so with a biopsy result as well. The Meerkat surprisingly reached to the conclusion of the yolk sac tumor, which is the correct answer in the USMLE question, because this is based upon the question, we know the answer, right? So that's good. And this is another example, we pass. And this is the question. And the correct answer is this. And this is the Meerkat's reasoning. The Meerkat reasons on each options, because the question has 4 options. Each option, the A, B, C, D. And the A is not the correct answer because of this reason. The B probably the plausible answer. C and D is not. So the conclusion is D. Conclusion B. And also some casual questions like a clinical query. Something like this. My eyes become red and itch whenever I visit my hometown. But my hometown and the city that I'm working in, they have a similar climate, and so what's the reason? There's no pollen present here, and the climate is same in both cities. What's the problem? How can treat it? And can you please recommend some drops? So the Meerkat suggests allergy testing, and also over-the-counter antihistamine eye drops that has ingredients like loratadine, and also mechanism of action of a particular drug, also a mechanism of disease. Cadasil is a rare disease. Wonder whether this Cadasil was explained in one of those 18 textbooks, but somehow it answered correctly. And finally, we will wonder whether the Meerkat that trained on the English textbook, the medical textbook, whether it can solve some Korean doctor license exam. So the cross-lingual question answering. This is one example of a Korean doctor's medical licensing exam, and this the first approach is zero-shot COT. Just give the Korean question to the meerkat. So meerkat somehow answer, in this example, particular example, is somehow answer correctly. In Korean question, English answer because we trained the English QA. The second approach is we translate Korean question into English and then give this English version. See whether this find the answer. Okay. The third approach is let the meerkat translate itself and then translate the English do the reasoning and then find the answer in English and then translate it back to Korean and then return to the user. So this approach is called pivot language guided generation. One advantage of this approach 3 compared to the approach 2. Approach 2 is you need to use external API for translation, like in this case GPT-4. That costs a lot. But the pivot language guided generation, you don't have to pay because we don't use API. The meerkat itself is translated. So it's quite desirable. And this is the result. GPT-4 by far is the best scoring LLM, even on the Korean medical license exam, 76. This zero-shot meerkat, 36. But after you translate and then give the translated English for the question, it achieves 66, and then let meerkat translate itself, and then solve the question. It's about 63. It's quite a promising number. So we'll keep working on this. This is one example of the plug approach. The Korean question translated version. The translation done by the meerkat, and then reasoning in English, and then back. Translate back to Korean answer. So this is the final answer. The answer is A. So this is the correct answer. So another example. So this is the final slide. Not sure whether some of you are aware of this. This is last year, September last year. This is about the story of a mother having a 3-year-old boy suffering from chronic pain. Visited 17 doctors over the 3 years, but did not find the diagnosis because it was a rare disease. You know, how many rare diseases in the world? At least 7,000. At least 7,000. Only those that we identified. There might be more than that. So it's very challenging task for one clinician, understanding all 7,000 rare diseases and then provide correct diagnosis. But quite disappointing. The mother logged into the ChatGPT. At that time, the ChatGPT was hot last year. The ChatGPT and then described the symptoms of his son. And the ChatGPT suggests some diagnosis, and then that was the correct one. So the boy is now happy. You need to know the diagnosis before you can treat the symptoms, right? So the LLM, this is only after the ChatGPT is slightly more than 1 year. So world has been changing, right? So the change will be even faster, right, from now on. So this story is one corner case, but it can be more common in the very near future. So we also are working on this problem, and hopefully we will contribute to this endeavor to get to this point. The LLM can contribute some the healthcare sector in overall. Okay. Thank you very much.